Good afternoon, everybody, and welcome to the imaging session in today's UK Biobank conference. We'll start off with just a few housekeeping notices. So as in the other sessions, please do feel free to put questions to the speakers using the chat function, and we'll ask these later during the panel. We'll have a 15-minute break at the end of the panel discussion. During the breaks, we'll be running the Early Career Researcher of the Year presentations. And as a reminder, all of today's presentations and slides will be available to you in the days following the event via the Engagement Hub. Okay, so on to the imaging session. And as you will all now be aware, the original UK Biobank cohort is 500,000 adults on whom extensive measures have been collected. The imaging extension to this is bringing back 100,000 of those individuals for brain, body, and heart MRI. And this imaging extension is by far the largest study of its kind worldwide and is game-changing in its scale. So the size of the study really brings unique power in the ability to identify early imaging biomarkers of future disease. So for example, from the 100,000 people being scanned now, we can expect relatively large numbers of them to go on to develop diseases pretty quickly. So for example, we'd anticipate at least 2,000 individuals to be diagnosed with dementia in the next few years. This then means researchers can go back to their brain scans in this case, which were acquired before the diagnosis, and see if there were any features on those scans that would have allowed us to predict who was going to go on and develop a given disease. And the same is true across all of the different imaging measures being acquired. So we can take the multiple measures of environment, lifestyle, genetics, etc., that are being acquired in the full cohort alongside the multi-organ imaging data and combine that information, for example, using AI methods, to learn which combination of measures relates to which long-term health outcomes. So that then means that in future individuals, you can use those combination of measures to predict their future long-term health outcomes. So that's the aim, and the schematic makes it look simple, but the reality of this obviously raises a number of different challenges. This slide here illustrates the complexity of the data set just here, considering the brain imaging data alone. This is just an example of some of the different metrics that are required in each individual, and the same is true of the heart and body MR. So this raises phenomenal opportunities. It also raises significant challenges. And some of those are going to be illustrated here, and perhaps we might return to some of these in the discussion points, and I imagine that some of the speakers might touch on some of these issues. So with the imaging data in particular, there are real challenges over how we go about creating useful measures, how we convert that raw imaging data into metrics that are useful for researchers, and what kind of information is useful to what type of researcher. Another challenge is how to leverage the phenomenal and unique power of this multi-organ data set. So typically, these different organs are studied separately. There's a real opportunity here to bring that multi-organ data together, but that raises a number of challenges, both in terms of analysis and in terms of interpretation. And finally, there are various challenges to us as to how to ensure that the results we get from this amazing resource are robust. There are statistical challenges around big data of this type that need to really be teased apart, particularly in relation to imaging data where there's lots of so-called researcher degrees of freedom. And then finally, I think the most important challenge is how we move from interesting observations that can be detected in these huge multidimensional data sets to actually having impact on health, whether that's for prediction, diagnosis, or intervention. So with that, I'd like to move on to the talks. The first speaker in this session is Professor Paul Matthews, who's the head of Department of Brain Sciences and the head of the UK Dementia Research Institute at the Imperial College London. And today, Paul will be talking to us about repeat imaging. Thank you.
Thank you, Heidi. It's wonderful to be with you all today. I miss the personal contact, but on the other hand, what a fabulous audience I've heard about. So let me just walk through, I think I'm going to remind you of the imaging enhancement, which is one of several enhancements you've heard about today to the UK Biobank data. This is allowing us to integrate quantitative, comprehensive imaging phenotypes from brain, heart, body, DEXA scans of the bone and carotid, along with baseline data, genetics, and past clinical history in order to begin to predict incident outcomes in novel ways. This is truly an exciting data set. At the time of the COVID-19 lockdown in mid-March, 48,765 data sets had already been achieved towards the goal of 100,000. We've all lost some time because of the lockdown, but the study will continue, and progress towards the 100,000 for sure will be made. Now, Heidi talked a little bit about what one can do with this. What we really want to, as a medic, what I'm interested in doing is understanding how quantitative trace can inform about the determinants of variation in the population, about the nature of aging, and of course, to define predictors of disease. This is some really nice work done by Wenjia Bai at Imperial, which has in which quantitative measures of cardiac structure and function have been extracted for the different chambers arrayed along each row across different measures of volume change running down each row, with each column being the different chambers. And what you can see there is across the population study, this is about 20,000, you can see the age-related differences in the population. That's hugely powerful data. One can go on from that to begin to relate these data sets acquired on individuals to all of the other data in Biobank that's been acquired. Along the abscissa, you see 11,000 individual pieces of phenotypic data from each of the individuals related to lifestyle exposures, comorbid diseases, and trace, and their relationship to these cardiac measures. This is opening up a really fantastically interesting discovery space for a multi-organ view of development, aging, and disease risk. Now, however powerful, the imaging working group, which I've had the privilege of chairing, has been excited about moving farther. So what we've done, what you see here, this is from a different study that is just used to illustrate the phenomena. This is a health rating scale by age with people studied at 2 time points across a large population. And what one can begin to see is that people's change in this particular measure over time define trajectories of behavior. Now, what we're doing in UK Biobank to date is we're taking a snapshot, just like the blue line straight down in this trajectory, or the red line for women, a snapshot that gives a cross-sectional view that does have explanatory power, some predictive power, but it's not as powerful as the trajectory itself. It's the change over time that can really tell us a great deal about individuals. Moreover, again, with the brain imaging, as Heidi alluded to with that beautiful picture of all sorts of fantastic colors arrayed on the brain, each of these imaging data sets, whether for the heart, the brain, the abdomen, the bones, or the carotid, contains an enormous amount of information. Again, for illustrative purposes, I've pulled this longitudinal data from the pioneering Rotterdam study. This is based on 1,000 people studied in the city of Rotterdam. And what you see here are the age-related changes across the population for people studied at more than one time, all stitched together across multiple measures simply from the brain. The top color bar, dark green, is a measure of fractional anisotropy in white matter in the brain, for example. The bottom brown curve that's so dramatic is the size of the ventricles, a measure of a measure of brain volume. What this is showing is there's a lot of change in these measures. They're highly sensitive to age-related changes. And on the right, you can see, I won't go through the individual measures, they're also highly sensitive to markers of potential pathology, all of which, amongst those chosen, are increasing with age. Now, the new frontier, of course, that a study like Biobank allows, that had really differentiated from previous studies, is the ability to relate changes in one organ to changes across others, and to really take this broad view of aging and disease in terms of interactions between and within organ systems. Again, for illustrative purposes, this is a different study, but it illustrates how powerful multiple measures, in this case of cardiovascular disease risk, are when begun to be seen in the context of adiposity across the body. Now, I've talked about relating, looking in detail at information across individual organ systems. I've talked about linking organ systems, both doing this over time. Now, if we start scaling up the numbers, we can begin to see heterogeneity in the population. Here, this is chosen from a particular brain example, looking at rates of change of brain volume, similar to that brown line I showed you earlier, over time in a population, and the outcomes data to the right, the probability of remaining free of Alzheimer's disease within a roughly 3-year period. And what one can see is those trajectories are powerful predictors, if you look at red, for example, versus blue, of outcomes within that 3-year period. Now, I'm going to unabashedly talk about re-imaging, the case for defining trajectories of individuals within Biobank using these sensitive quantitative tray markers. I'm going to unabashedly talk about this in terms of my own interest, which is dementia. And Biobank has shown itself already with the cross-sectional measures alone as a powerful resource for dementia research in defining genetic architecture of the brain, environment and lifestyle risks for brain health and aging, understanding some of the risks conferred by multimorbidities and defining them in quantitative ways, and then very, very important, beginning to identify factors that may change long before there's clinical expression of the disease and be more powerful predictors than is otherwise available from the clinical phenotypes. I believe, again, with my unabashedly clear interest in dementia, that Biobank, with re-imaging and the ability to define trajectories of quantitative tray markers, can make a powerful contribution to the major social challenge of dementia. Not long ago, the Alzheimer's Research UK produced a very powerful statement delivering the dementia moonshot, setting an agenda for a prospective longitudinal monitoring and biosample collection from a large, initially cognitively healthy cohort in which there could be assessment of the genetic, environment, and lifestyle risks, and a broad range of advanced approaches to characterizing the earliest pathology could be deployed in a way that would allow linkage to clinical records for the study of comorbidities, and finally, that all of these data would be open for use by researchers worldwide. Now, it wasn't stated, but frankly, this is what UK Biobank is becoming and will be. Already, re-imaging has been initiated with tremendous support from Dementia's Platforms UK and its lead, John Gallagher, funded by the MRC. It's put in place initial funding for re-imaging of 10,000 participants, all of which would have a repeat of the original examinations they underwent with this interscan interval of 2 to 2.5 years. what's extraordinary is after people go through this rather significant protocol, 88% of the people have all said that they would come back for the re-imaging if asked. Up to the time of the COVID-19 hold in March, over 3,000 people had already completed their repeat studies with a mean interval of 2.25 years. So we're well on the way to that 10,000 participants. Unfortunately, fully a tenth of that group will develop dementia by 2026, meaning that we're well within, we will be well within that window that I described in the trajectories earlier that shows how predictive, in that case, brain measures could be. Now, that's exciting, that's going to be important, but the imaging working group has even greater ambitions. And I'm pleased to say that recommendations from the imaging working group to Biobank were endorsed 2 years ago by the International Scientific Advisory Board, and the planning was initiated to begin a broader re-imaging program to be initiated when the current scanning is completed, using the infrastructure and staff that are already in place, with the objective of getting 60,000 additional repeat assessments, reducing the number of scans per day so that one could have enhancement of the imaging protocols, adding cognitive assessments, and improving completion rates, extending the interscan interval for predictive value up to 7 years. And what I'm really pleased to close this talk on, and today in part, is that UK Biobank is already in advanced discussions with a funding consortium that I hope is going to be able to announce very soon that this can go ahead. Finally, I want to close with a point that I hope everyone has realized throughout this exciting day of science. UK Biobank and all that it does, the imaging enhancement being no different, are huge team efforts. And I want to acknowledge the great commitment of UK Biobank participants, the staff of UK Biobank, the members of the imaging working group, and for us, more than 125 people around the world who have contributed to the design and to helping us solve problems as we've gone through. Thank you.
Thank you, Paul. Really exciting to hear about the prospect of potentially even more repeat imaging being added, because you showed very convincingly the added power that you get from those repeat measurements. So we'll move straight on to our next speaker, but just to remind you, you can use the, please do post your questions to the speakers and we'll come back to them at the end. So our next speaker is Professor Carla Miller, who's Professor of Biomedical Engineering at the University of Oxford and Associate Director of the Wellcome Centre for Integrative Neuroimaging. And she is going to be talking to us about the brain imaging measures. I think we have a slight technical hitch, so we're just trying, apparently, we're just trying to reconnect with Carla, so sorry about that. We will move as soon as we're able to Carla Miller's talk.
All right, have we got connectivity yet?
Yep, go ahead, Carla.
Okay, brilliant. My apologies about that, I'm not sure what happened. So I'll just begin from the beginning. So as Paul mentioned, all of the imaging enhancement is a huge team effort. But in particular, the brain imaging, I think, has done something quite remarkable, which is to take the kinds of studies, very multimodal studies that we would typically require something like a half an hour to acquire all of this data and to pack that into a very dense 35-minute scan. And what we get from that is the ability to have very rich multimodal data. So we have 6 different imaging modalities in the brain imaging, which enables us to, in the case of brain structure, we have 3 different modalities which enable us to segment the brain into its component structures and to estimate their volumes and thicknesses of tissue. We have the ability to using T2-weighted imaging to identify white matter lesion load, which is relevant to a number of diseases and is a common factor in aging. And we also have quantitative measures from MR, including T2 star and quantitative susceptibility values, which reflect things like the tissue constituents such as iron and calcium that are very relevant to disease. In terms of the gray matter in the brain, where brain activity occurs, we have 2 different types of imaging, including task fMRI, where we are able to, where we have subjects performing a cognitive task while they're being scanned. We also have what are called resting state scans, where we look at spontaneous fluctuations. So this enables us to look at the amplitude of activity in certain brain regions, but also to identify connections in the brain, so how different regions are communicating with each other. In terms of the brain's white matter, the diffusion MRI measures that we get are particularly relevant, and this gives us 2 types of information. It tells us about the pathways that form those connections in the brain, but it also tells us about microstructural properties. So although our pixels in MRI are typically on the order of 1 millimeter, the signals themselves can be sensitive to the tissue structure on the order of micrometers. And so this gives us cellular level pathologies. So having designed this relatively complex multimodal data collection protocol, the next priority for us in the biobank brain imaging was to develop a pipeline for analysis that can take us from the raw images, which are not particularly informative, all the way through to imaging phenotypes that non-imaging experts can use. And so this is work that's been led by Steve Smith's group, and in particular, Fidel Alfaro Almagro has had this as his PhD project, and he's designed a very impressive data processing pipeline. This kind of processing is very expertise intensive, and so you really do need imaging experts to push this forward. But what Fidel's managed to do is to take these raw images that are difficult to interpret and produce both images that are in of themselves more interpretable, partly because they're aligned across subjects, but also because the actual pixel values are meaningful. But he's also managed to turn these into what we call imaging-derived phenotypes, or IDPs. And these are just scalar values that can be used much like other phenotypes in other biobank measures. Having developed that processing pipeline that takes raw images and produces phenotypes, the next high priority was to come up with confounds. So imaging is not as specific as we would like it to be. This means that, that there are a number of types of confounds that can affect our imaging-derived phenotypes, or IDPs. So for example, head motion can create image artifacts. And these artifacts can then creep into our IDPs. So if you're studying a disease where patients may be more prone to moving their head, then you would want to know whether a given association is primarily driven by this artifactual head motion, or is it actually more biologically interesting than that? So Fidel Alfaro Almagro and Steve Smith have created a rich set of confounds that they have carefully vetted as being relevant to biobank IDPs, and they've generated a sort of a standardized set of confounds that can be distributed alongside the IDPs for people who have less imaging expertise or background to work with. And many of these, like head motion, are well known in the imaging literature, but there are actually other confounds that they've identified that really speak to both the power but also the complexity of the biobank imaging data set. And I'd like to give you one example of that. So this is a confound that that they have derived, where they took a large number of imaging phenotypes across a large number of subjects, and they identified they ran some analyses where they identified principal sources of variation across those phenotypes. And in this case, this is showing a clear effect of time of day, from morning on the left to afternoon and evening on the right. And not only do you see a drift in imaging-derived phenotypes, which we might have expected, but in addition, there's an interesting oscillatory pattern here. Now, in terms of understanding what this pattern relates to, we can overlay this on top of the time of day at which the number of subjects that are typically being scanned at any given time per day. And you can see that there's a very clear pattern here where these oscillations correspond to triplets of subjects. And this makes a lot of sense, as individual participants will come in to the center, the imaging facility, as a triplet, and as they simultaneously enter, different subjects will be going through the imaging stations in a different order, and this clearly has an effect on these imaging-derived phenotypes. But this is an incredibly subtle effect, and not one that you would expect to find in a more conventional study with a lot smaller number of subjects. But it is the kind of effect that we need to be able to capture and account for if we're going to be able to have sensible and clear interpretable associations. So this is the work that we've been doing on behalf of the UK Biobank. I'd now like to move on to some of the studies that we've been conducting as Biobank users. And the first types of studies that I'd like to talk about are just the classic association studies. So this is one of the first studies that we as Biobank users published in 2016 on the first tranche of subjects, where we had at that point 2,500 imaging phenotypes, which we then did pairwise associations against 1,100 non-imaging phenotypes. So that's 2.8 million association tests. But due to the powers of numbers, in this case 5,000 subjects, even with multiple comparison corrections, we ended up with 30,000 significant associations. And you can also see here that it's not confined to one of the imaging modalities, but all of the imaging modalities are throwing up different associations, really suggesting the power of this multimodal imaging approach. And note that the log P significance values here are on the order of 50. If we now look at what this looks like for 40,000 subjects, we see that with this larger number of subjects and indeed an enhanced set of phenotypes, now 4,000 rather than 2,500 imaging phenotypes, we're getting just under 500,000 significant associations with log P values on the order of 300. So as the resource continues to grow, we're able to identify a richer set of associations. Now, of course, as the imaging data came online in this should have a reference to a paper in 2018, we were able to then perform some of the first imaging GWAS studies. In this case, we had 8,000 discovery subjects, which we did pairwise associations against 11.7 million SNPs. And here, what we did was to identify clusters where we have sets of imaging-derived phenotypes alongside sets of SNPs that tended to cluster together and which were replicable. And in 2018, when we published this first paper, we provided also this brain imaging genetics, or BIG server, where we provide the summary statistics as well as have a browsable view of the data for people to explore on their own. Now, the update to this in 2020, with the most recent release of 40,000 subjects, we end up with, and including the X chromosome, we now end up with about 600 clusters which replicate. And we've recently put together an updated version of this server where people, again, will soon be able to go and browse the data, but the summary stats are already up on that server. So just to give a feeling for what some of these imaging hits look like, this is one of the imaging hits that we identified, where we took an imaging marker, which was T2 star in the putamen, so this yellow structure that you're seeing, and we identified about 4 different peak SNPs that showed significant associations with this particular imaging measure. Now, in order to try and understand what these associations are telling us, we can start to dig a bit further into this data. So in this case, if we take each of these SNPs, and my apologies that the slide has been a bit corrupted here, so that the colors in the circles are supposed to map onto those peak SNPs and correspond to the images on the right. What you can see here is that although we identified these SNPs as having an association somewhere in the putamen, as you go into taking each of these SNPs and regressing it at a voxel by voxel level back into the images, that you get a more complex picture. So each SNP is subdividing these structures up in a slightly different way, and in some cases it's pulling in additional structures. If we then look into what the associated genes appear to be, so one of these peak SNPs lies in a portion of the genome where there are genes associated with iron transport and storage. And this is quite an interesting output, given that these T2 star measures that these associations are derived from are imaging markers for tissue iron. So this is clearly pointing us at a consistent story biologically. So I'd like to change tack a little bit and talk about a few other approaches that we can take that are not sort of discovery studies like association studies. In this case, this is a basic science study that we published last year, where we wanted to ask the question, does brain microstructure predict brain function? This is something that we know from animal studies should be a consistent relationship, but it's actually not been demonstrated in brain imaging data, despite these basic modalities having been around for quite a few decades now. What we did was to take the brain activity measured by functional MRI in the gray matter, specifically looking at the degree of coupling between pairs of regions, and ask whether we can predict that degree of coupling purely based on properties of the white matter pathways that connect them. So whether those pathways that connect them predict the connectivity of the brain activity itself. And what we were able to find is that across a large number of brain pairs, so nearly 80 different brain regions, not only were we able to predict a reasonably large fraction of the across subject variation in functional connectivity, so on average about 3%, but as much as 10% of the cross-subject variation in functional connectivity, but that this was highly reproducible. So when we applied a model fit to an 8,000 set of subjects, when we blindly applied that to unseen subjects, we got a very consistent pattern of the amount of functional connectivity that we could explain. In order to interpret this, we can then take these relationships between microstructure and function, and we can use these as regressors in an imaging GWAS. So in this case, what we identified was across several different brain regions was one gene called DAM1, which is part of the Wnt signaling pathway and which has been implicated for axonal guidance and growth, the precise cellular components that actually form connections in the brain. So again, it provides a very clear biological picture. The last set of type of study that I want to tell you about aims to use the richness of the biobank data in order to define data-driven phenotypes, which provide a more complex view onto a target feature of interest. So the target feature of interest could be a disease or a condition, but in this case, we aim to use age. So this is something that's been taking off in the brain imaging literature, and it's often referred to as brain age. The idea is to take brain imaging features and use them to try to predict a subject's age. So here you can see that by doing this, we can form quite a good prediction. On average, we have an error of slightly less than 2 years in terms of brain predictions. But more interesting than our ability to predict age is to understand what happens when we don't predict age very accurately. So in this case, if we identify a subject which is that deviates from this population norm, we can define a brain age delta. And my apologies that the delta here is not quite displaying properly. But the idea is that a deviation from between the predicted age and a subject's actual age indicates that their brain is aging faster or slower than the population norm. And that is potentially a biologically interesting effect. So in conventional brain age modeling, what you would do is take imaging data, you would extract some imaging features, and you would use that to form an age prediction. And a key point of the standard brain age literature is that you would form a single age prediction. You would assign to a subject one brain age that may deviate from the population norm. And what we were interested in doing was moving away from a single brain age prediction and asking whether we could form multiple brain age predictions under the idea that different tissue types or brain regions may age at different rates, and that would be biologically and clinically interesting. So what we wanted to do then was to identify multiple modes of brain aging. And the way we did this was to use something called independent component analysis, which takes the imaging data and it tries to explain variance across a range of different modes. So here I'm showing you a series of modes, which was derived.
Wrap up in the next minute or two, please.
Yep, no problem. Yeah, so these different imaging modes are derived in a data-independent way, but as we see in this curve here, a lot of them do actually have a strong age correlation. So in this mode, for example, we see a brain aging mode that seems to relate to the bone marrow, and which shows sex differences. In particular, we see that females have a sharp trajectory that changes around the age of menopause. So that's one example mode, but this is my last data slide, where I guess a big outstanding question is whether these modes can really be demonstrated to have biological meaning. And I think that's shown very nicely, where here on the top, we're showing imaging GWAS using modes where we identify a rich set of associations across the chromosome. But on the bottom, if we have a single brain age that we predict, many of those associations go away, suggesting that we've diluted out the richness of information that we have here. So with that, I'm going to conclude. I'd like to particularly thank Steve Smith, Paul Matthews, and Fidel Alfaro Almagro, who have been huge in leading the imaging GWAS, as well as all of these individuals here. And thank you for your time.
Wonderful. Thank you very much, Carla. And now moving away from the brain for our final speaker is Professor Daniel Ruckert, head of the Department of Computing at Imperial College London, who is going to be talking to us now about the cardiac imaging.
Thank you very much. And I'm going to follow off where Carla actually left off and try to show you something very similar with the cardiovascular imaging in Biobank. So I'm going to really focus very much on the cardiac data, which is dynamic data acquired as part of UK Biobank, which of course can also be analyzed exactly in a similar way to how the brain imaging data is analyzed in Biobank, by identifying correlations, for example, with lifestyle, genetics, or of course, also with health outcomes. But before I show you some of these examples, I wanted to also show you how we address one of the other fundamental problems in the imaging data in UK Biobank. And that is, of course, we have acquired already a huge number of these images. And it's amazing to see nearly half of the data already being acquired. But we also need to make sure that the data we have acquired is actually suitable for analysis. So one of the first questions we asked is, can we actually use artificial intelligence to tell us whether the images we are acquiring can be actually analyzed automatically, because we don't have any other option of analyzing the data? And of course, is the data of high enough quality, and can we identify any potential problems with the acquisition? And what I wanted to show you here is a piece of work which we've been doing in trying to implement different image quality checks. Cardiovascular imaging is perhaps a bit different to the brain imaging in a sense that there are many sort of subject-specific things which can go wrong. We need to cover enough slices through the heart to actually cover the heart in 3D. Typically, every slice is acquired in a different breath hold, and we need to make sure that the subjects are complying with the instructions so that we don't have too much interslice motion between different slices. And of course, there are a number of reasons why contrast, for example, might not be as good as we expect it to be. So we've developed an automated machine learning pipeline which estimates all these parameters directly from the data. And what you see here is, for example, two examples of data in UK Biobank. The first row you see is high quality data where, for example, the left ventricle has been covered by more than 100% with slices. So that means we can actually do a proper three-dimensional analysis of the anatomy of the left ventricle and its motion. There is very little interslice motion, so the data is actually consistent if we stack it together in 3D, and also has very good contrast. On the bottom, in the bottom row, you see a data set where there is actually only part of the left ventricle covered. So we're missing out here both the apex and the base, as well as we have problems with slices being misaligned due to the fact that the patient or the subject has been holding their breath in a different, in an inconsistent position. And you can also see that the contrast is not particularly good. One of the great things which we can report is that actually the data in UK Biobank, the cardiovascular imaging data, is of very, very high quality. We have, for example, looked at the percentage of coverage of the heart in all of the data which has been acquired. And as you can see on the left-hand side, nearly all of the data has been acquired with 90% plus coverage, in most cases really 100% coverage or more, which is very good. And in addition, you can also see that the gaps both at the apex and the base of the heart are very minimal, meaning that actually we have really all the necessary data to do a 3D analysis. The other thing which we can, of course, do is we can also provide feedback to the image acquisition. So what you see here on the left-hand side is the amount of coverage of the left ventricle at two different imaging sites in UK Biobank. And on the right-hand side, you see a similar plot showing you the coverage at different dates of the acquisition. So, and you can see that, for example, the beginning of the acquisition, I think we're probably pushing too much for time, and therefore there is less coverage of the left ventricle than in subsequent image acquisitions, where we could then use that sort of information to provide feedback and decide that we want to, for example, slightly change the way we do the imaging. Another aspect which is quite critical for 3D analysis of the heart and the derivation of image-derived phenotypes is to look at that the data is consistent in 3D. And as I said before, the data is often acquired with different slices are acquired in different breath holds, meaning that the subject has to really hold their breath in a consistent manner to allow 3D analysis. And what you see here on the left-hand side, we look at the average misalignment between slices, and you can see it's very minimal. And both at the apex and at the mid-ventricular slice, as well as a basal slice, we have very good consistency. So the data really is of very high quality, and it's very good that we are able to automate this assessment of the data. Obviously, one of the keys we want to do with the data is to derive phenotypes from the imaging data. And Carla showed you already a very nice pipeline for brain imaging data. And it's probably fair to say that the analysis of brain imaging data has been a bit more mature than the cardiovascular image analysis. So we have developed a lot of tools specifically for the analysis in UK Biobank. One of the approaches we have taken is very much to take a machine learning driven approach, where we take data which has been manually annotated and then train a machine learning, a deep learning algorithm to do this automatically. And what you see, for example, here is that we are able to automatically segment the different chambers of the heart, the left and the right ventricle, the myocardium, as well as the left and right atrium in a fully automated fashion. Now, I won't bore you with all the details, but of course, one of the important things in the heart is that all of the, or much of the data which is acquired is dynamic data. So we're not only interested in analyzing the anatomy and the morphology at a given time point, for example, the end systolic time point, but we're interested in tracking anatomy throughout the entire time series, because that tells us something about the function. And we've been developing several tracking algorithms to look at dynamic structures in the heart and being able to localize them in every frame of the sequence. For example, here you see the ascending and descending aorta, which we can then fully automatically track through the imaging data, even if if some of the frames are of less good quality. So all of that, putting that together, gives us a pipeline where we can look at the anatomy of the heart, the morphology of the heart, of the great vessels as well. And in addition, because we can track the information throughout time, we can also look at parameters such as wall thickness, as well as motion parameters of the heart. So we can quantify parameters such as the radial and circumferential strain from the imaging data. And that then gives us a whole set of image-derived phenotypes, exactly as we have in the brain, which we can try to analyze. For example, here you see some normative values extracted from over 25,000 subjects in UK Biobank. We've stratified them into men and women here. And obviously, there are size differences in the cardiovascular system between men and women. But of course, we can also correlate these with other parameters, such as, for example, with aging. And what you see here is a quite nice plot, similar to what Paul Matthews has shown you in the beginning, looking at, for example, age-related changes. And what you can see if you look, for example, at the bottom row, we see the ascending, descending aorta, and we can see that the area of the vessels is slightly increasing with age, but the distensibility, so the elastic nature of the vessel is lost with aging, and is reduced as we get older. So we can now do all of these association studies. We can, of course, also look at, for example, regression coefficients, which relate these cardiac and aortic phenotype to, for example, demographic information, which are these blue circles you see here in the plot, to anthropometric information, which are the green circles you see in this diagram, as well as to cardiovascular risk factors. Of course, we can also relate these to common diseases. What you see here is a sort of correlation of different phenotypes derived from the imaging data in UK Biobank to common diseases. And of course, there are many of the common diseases we expect to be highly correlated to cardiovascular parameters show up here. But of course, it's also very interesting to see that in some cases, there is still even in less obvious cases, there is a correlation, even though that correlation or strength of association is less than we expect. Showing you now a very similar analysis to what Carla showed in the brain, a sort of phenome-wide association study, looking, for example, at the p-values between image-derived phenotypes of the heart and non-imaging information. And of course, because we're looking at the cardiovascular system, the measures are particularly strong related to physical measures. And here I'm just showing you the same information using the strength of correlation indicated by the size of the circles you see in this particular plot. If you wanted to explore this in more detail, of course, the data is available within UK Biobank, but you can also access an interactive visualization platform at this web address if you wanted to interactively query the data and look at certain image phenotypes. You can explore this interactively. And perhaps just to conclude here, I wanted to also show you some examples of where we're planning to go next. So at the moment, we're at a fairly well-defined number of phenotypes, around 80 phenotypes. What we're planning to do in future is much more look at the regional structure of the heart and how we can derive regional phenotypes of the heart. What you see here on the right-hand side, for example, is a surface model of the right ventricle. And the spaghettis you see superimposed on the surface show you the motion trajectory of every individual surface point on the right ventricle. And if we take that into, for example, a machine learning model to try to predict risk, or in this particular case, we're predicting survival in patients with pulmonary hypertension, we can identify that these dense phenotypes, which we can derive on the anatomy of the heart, can be much more powerful than global descriptive measures, which we derive from the heart at the moment, and give you a much better separation between different population cohorts. And with that, I wanted to just thank all the colleagues who have been helping us with doing this. And as many of the previous speakers said, this is really a team science sport. And I'm very grateful that we had such a fantastic team helping us with this work. Thank you.
Speakers for really excellent, stimulating talks. We are going to move now to the Q&A session, so hopefully all of the speakers will reappear. Thank you to everybody who has been sending in questions. We will get through as many of them as we can in the time available to us. So to start off with, I might start with what's hopefully a few quick and easy questions for Paul Matthews to begin with. So Paul, if you're there, the question is, when will the full imaging dataset be released for use by the research community, and when is the longitudinal MRI data or parts of it available?
Thanks, Heidi. The data set is being released serially, roughly each quarter. So the last release had about 40,000 individuals included in it. The first, I'm embarrassed to say I am not sure of the schedule for the release of the re-imaging cohort. And if the individual interested wants to email me, I can make sure that we get information on that to them. Carla tells me that 1,500 people have already been released out of the slightly more than 3,000 who've been scanned. And I'm sure we'll see a further release in the next tranche, which will follow the reinitiation of imaging post-COVID.
Wonderful. And while you're on the subject of that, another quick question is, when do you expect to reinitiate imaging post-COVID?
Planning is already underway, but I can't give a specific date. As everyone knows, things are changing in the COVID lockdown on a daily basis. We've just heard positive news from the government over the last hour, and I'm hopeful that things can move more quickly as we move to the end of the summer and into the autumn.
Wonderful, thank you. Carla, interesting question for you. Can you explain why time of day has an impact on the IDPs? So, for example, on the brain imaging measures that you showed.
Yeah, it's a really good question as to why exactly that effect shows up. I think the most intuitive explanation is that it relates to data quality as people are moving through in a very high throughput situation, that over the course of the day, as things get a bit more hectic in this very high throughput situation, that it's likely the data quality is just being impacted. But I don't think we have a very clear idea. It could be also, for example, that so if you think about these sort of oscillations of IDPs, the third, where we have these triplets, the third person so triplets of people who are moving through the imaging center together, the third person in that triplet has just been through 2 previous scanning sessions. So it could be that at that point they're getting a bit tired of being in scanners and are more likely to be moving or something like that. But we don't have a very clear answer to that question, I'm afraid.
Okay, thank you. Daniel, you showed interesting work that your group have been doing on segmenting the cardiac images. Do you have any plans to apply similar approaches to segment organs using a similar methodology?
Yes, and that's a very good question. I think on the cardiovascular data, I have to say we were extremely lucky to have collaborated with Stefan Peterson's group at QMUL and also with Stefan Neubauer in Oxford, who have actually provided a lot of manual annotated training data, which we could use to actually train our algorithms. And that has meant that we can roll out very quickly on the cardiovascular side. We're basically at the moment trying to train our algorithms exactly with organ segmentation for UK Biobank. But of course, there the process, the real bottleneck is acquiring the training data, which we have to do at the moment. And I hope that we will be able to basically within the next 3 months release organ segmentations for much of UK Biobank, using very similar approaches, which are sort of using training data from experts and then sort of rolling it out directly in UK Biobank.
Okay, thank you. And while you're discussing the tool, so another question for you is the QC software that you presented open source?
Yes, it is open source. There was a reference on the slides which shows the paper, and you can also get the software. I have to admit that the software is obviously very much geared towards UK Biobank data. So we have really fine-tuned it for UK Biobank data, but it can also be used with clinically acquired data, as long as you have the similar sequences as in UK Biobank.
Okay, thank you. Paul, another couple of questions on the plans for repeat imaging. There's a question as to whether the proposed 60,000 with planned re-imaging will also include repeated cardiac MRI, and also whether you'll be taking cognitive measures alongside the repeated brain scans.
Yes, all of the same imaging measures that were included in the baseline imaging will be repeated, including the cardiac. We hope to be able to take a little bit more time in the imaging as well, which may improve some quality in the cardiac scans even further. Cognitive measures are very, very important. They were acquired throughout UK Biobank already, but we hope that there will be some enhancements in these with the addition of perhaps one more test in-house, and perhaps also the use of an online test, another online test for people at home before they come in.
Okay, thank you. Thanks, Anna. Carla, a follow-up question for the brain imaging about the fMRI. Could you outline what are the fMRI tasks that are included, and what's the sample and age range for the fMRI brain tasks?
So the tasks, there's actually only a single task, which is referred to as the Hariri task. So it's a very stripped down version of a quite well established task where subjects distinguish between they either have to distinguish between different shapes or between different faces, and it was chosen basically because it would elicit activity in a range of different brain regions, all the way from basic sensory up to some of the limbic system, where these faces are quite emotive. There's details about that on the Oxford website describing the imaging resource, which people are very welcome to email me about. And then there was a question about the age range.
Mm-hmm. Yeah, what's the sample and age range?
Yeah, so I mean the age range, I don't know exactly, but if you take the original Biobank study, we're 5 years on from that. So I believe we are something like we're either 50 to 75 or 45 to 70. But of course, as it's taking us a while to acquire all of this data, that age range is actually expanding a bit.
Okay, great, thank you. Question, I'm not sure who to, but can anyone comment on IDPs for liver images?
Yes, I'll say a word about it, Heidi, very briefly. So there's been an exciting partnership with Perspectum, a startup company out of Oxford, and with Stefan Neubauer and his team, to develop a set of protocols for fat quantitation and for iron quantitation within the liver. The fat quantitation metrics are available as downloads from UK Biobank, and there's descriptive material available in the published literature associated with UK Biobank. There is opportunity for enhancement of that data going forward using a variety of methods, and we look forward to seeing lots of people studying the liver. It's a rich source of phenotypes.
Wonderful, thank you. Daniel, interesting question for you. Are there efforts towards interpretable AI for analyzing these heart data? That is, making the results obtained using machine learning understandable by humans, which could have potential implications for treatments?
That's a very good question. So at the moment, almost all of the image-derived phenotypes we extract are measures which you would normally, which perhaps in radiology or in cardiology you would normally acquire. For example, measures such as ejection fraction, so the amount of blood volume the heart can pump. And almost all of these parameters are derived from segmentations of the heart. So they are inherently interpretable in that sense that they are that they're visually verifiable. But of course, what I showed at the end is where we go into much more richer phenotypes, where we, for example, look at every voxel or every point on the heart surface and how it moves throughout the heart, and then actually put that into, for example, a machine learning model, a deep learning model, to derive a new image phenotype. Then, of course, developing this AI, explainable AI solutions is really, really important. And many of the methods which we work on are at the moment very much focused on what we call attribution methods. So they show you visually which voxels contribute to a certain prediction, whether they contribute positively or negatively. But of course, that can be quite difficult sometimes to interpret. So converting that into something which is biologically meaningful is not an easy task, and I think can only really be done via sort of a team effort between those developing the AI tools as well as those who are experts, for example, in cardiovascular morphology function, who actually can also give some sensible interpretation to the data.
Great, thank you. Question for Carla. Have you released open code or software for the brain age estimation for people who might want to try it out on non-UK Biobank data sets?
Yes. Yeah, in general, we basically always release all of our code. And in the papers, there are links to those sources. Yeah.
Okay, great. Thank you. Question, I don't know if anyone knows, what about IDPs for eyes? IDPs, as in...
Yes. So there was one of the earlier enhancements for, and in fact, arguably the first imaging enhancement was for optical coherence tomography. And that data is available within UK Biobank and already, and there have been a number of exciting papers published with more coming out.
Great. Wonderful, thank you. And Paul, another question on the repeat scans. How will the subjects for the repeat scans be selected?
Our hope is to move up to the 70,000 target, which is really approximately the achievable number given the number who said that they would come back and the attrition. So in that respect, the ambition is to repeat as many as it's feasible to. For the first repeats, it's been targeted around those who had the first scans, which were centered around Cheadle outside of Manchester.
Okay, great, thank you. Interesting question here that could be for any of you, but maybe I'll direct it at Carla to start with. So for researchers that have less experience in imaging, is there an official guide to using these IDPs so that the effects of confounders are minimized?
Yeah, this is a really hard question. So I think, one of the things that I think we recognized very early on is the value in providing IDPs so that people don't have to suddenly become imaging experts. But it's really difficult to come up with a single answer as to what a confounder is. So, for example, there are plenty of situations, I talked a bit about age, there are plenty of situations where age might be a confounder and other situations where age might be an effect of interest. And I think this is where a bit of domain expertise really does help, is to understand when an effect like that might actually act as a confound. So for the work that I described, where on the brain side we have been developing a standard set of confounds that we distribute, I think there are sort of our suggestions of best practices. But at the end of the day, I think there's not going to be a sort of a straightforward answer of always do things the way that we've suggested, and it's always going to be the right thing to do. So I think there are best practices that are starting to come out, and I think people should read those over. But then I think it's never a bad idea, if you're going to be working with imaging data, to try to get some kind of imaging expert in the loop who might be able to kind of identify some of the gotchas that are sort of well known in the imaging community, but not necessarily so well publicized.
Yeah. Okay. Great. And I think probably final question to Daniel. So are your derived IDPs publicly available, and if so, where?
Yeah, they're all available in UK Biobank, and they can be downloaded from UK Biobank.
Okay, great. Well, that was a very quick answer, so we can probably fit in another quick question in that case. Paul, will the DEXA scans be repeated as well?
The intention at this point is to repeat the DEXA scans, and it is with DEXA in mind, as well as cardiac, that we're particularly interested in looking at the longer time intervals.
Okay, great. And then a final quick one for Carla. Are IDPs available for the cerebellum?
There are some IDPs available for the cerebellum. Again, if you go to the Biobank showcase, it's relatively straightforward to have a look and see what's there.
Okay. Wonderful. Thank you all very much for excellent sessions, really fantastic talks, and lots of useful discussion points. I think I now will just be handing back over to Rory Collins, the PI of UK Biobank, who's going to be announcing the winner of the Early Career Research Prize. Thank you.
Thank you very much. So we had over 70 submissions to the Early Career Researchers application from all around the world. And the two runners-up have presented in the previous gaps between the sessions. And it's a great opportunity to congratulate them, Connor Ebden from the Broad Institute in the US on his study of clinical hematopoiesis and the risk of chronic liver disease, as well as Katie Young from Exeter on HbA screening in UK Biobank and detecting undiagnosed diabetes. Given the kind of international nature of the submissions, it's quite encouraging from the perspective of the quality of scientists around the UK, that the winner is also from the UK, Leyland Taylor from the Sanger in Cambridge, who's going to talk about a very appropriate final talk after the imaging session, because he's going to be talking about epidemiological and genetic analysis of DEXA imaging data and what he has revealed with it. So I'll now hand over to the video from Leyland, and my congratulations to him. It was a very competitive field.
Hi, my name is Leland Taylor, and it is a real privilege to be here today to talk to you about abdominal aortic calcification in the UK Biobank. So vascular calcification occurs in the intimal or medial layer of vascular tissue. It is commonly observed in patients with hypertension, diabetes, or chronic kidney disease. It is associated with cardiovascular morbidity, yet its pathophysiology is poorly understood. This is in part because it's just poorly studied, as measuring it involves a time-consuming and manual scoring process. So the UK Biobank imaging study features low-intensity X-ray scans, and from these scans, one can calculate a variety of kind of standard phenotypes, including bone mineral density and bone area. And what we found is that you can also identify abdominal aortic calcification from these images. So in this study, we built a model to automatically quantify AAC from low-intensity X-ray scans. Using that model, we quantified AAC in nearly 30,000 UK Biobank participants. And then we conducted the largest epidemiological and genetic analysis of AAC to date. So there's a lot of findings that we have, and you can find the full details of all of them inside our medRxiv preprint. Today, I just wanted to highlight a few of the key findings. First, we found AAC was relatively common and detectable in 12% of participants. Second, we found no evidence of an association between LDL levels, serum calcium, or vitamin D levels, things you might have thought would be associated with aortic calcification. Third, we found AAC was associated with a variety of phenotypes. Most notably, it was associated with increased risk of myocardial infarction. And was just I would here. Yeah, from
Thing
Fine. And
eat
and reason. Doing all of you.
to really are you we Today we have the compute the we would have load these have used